All right. The blue strands of DNA on this diagram are our original parent strands. So they are called template strands because they do form the template for a new DNA strand being formed. When we're going to have DNA copied, it's called DNA replication. And the first thing to happen is that this double helix needs to unwind and be separated in spots. So the enzyme helicase is responsible for unwinding that helix. It doesn't unwind the entire strand. Remember, you have 6 feet of DNA in your cells. It's not going to unwind all 6 feet. That would be a big tangled mess to deal with. It only unwinds small portions and deals with small portions at a time and then moves on to unwind the next portion. That's why DNA is called semi-conservative, because it's not undoing the whole thing all at once. It's only doing it in small pieces. So here's our small piece, and the junction point where it's the unwinding and wound DNA meet, that's the replication fork. So that's the point of replication. DNA polymerase, another enzyme used in this process, reads the DNA strand and pulls in the right nucleotides from outside to build this new strand of DNA photoed in red here. It is going to, pause that. All right, so DNA polymerase is going to read the template strand, bring in the right nucleotides to build the complementary new strand, and the DNA strand that's formed happens to be half old, half new. Still semi-conservative. Okay? It reads that strand from the 5' to the 3' ends, and the other strand, since that's going the opposite direction, it will break it up into smaller chunks called Okazaki fragments. Later, another enzyme is going to come by and join those fragments together. And the next step after all of that is DNA ligase is going to bind up the whole helix again. Lig means to bind. Those of you that play clarinet or saxophone, it's your ligature that holds your reed to your mouthpiece. In CSI shows, you've heard the coroners talk about ligature marks. Those are strangulation marks on the necks of strangulation victims. Lig means to bind, and so ligase is the enzyme that's binding up our brand new DNA. What on problems 14 through 21 did I not cover?